Good evening, ladies and gentlemen. Welcome to our third public forum for this year. The theme that we have set up for this year is Understanding Cancer Research and Current Treatments, Empowerment Through Knowledge. Bonsoir, mesdames et messieurs. Bienvenue à notre série, le troisième de notre série de forums publics pour le Centre de cancer Goodman. Le thème de cette année est Comprendre la recherche et les traitements actuels du cancer, le savoir au service des patients. Tonight, we will be discussing a very special type of cancer, brain cancer. The latest research advances in the new treatment strategies. We have an all-women team to discuss this. My name, if you have forgotten, is Nicole Beauchemin. I'm a professor in the various departments here at the university. I'm a full member of the Goodman Cancer Research Center, and I've been moderating these forums for a number of years. I'm happy to see some familiar faces, and I hope that you learn quite a bit from our experts. Our first expert is Dr. Nada Jabado. We go back a long way. Nada is professor of pediatrics and human genetics here at McGill, and she essentially has her laboratory and her clinical duties at the Research Institute and at the McGill University Health Center here at the Glen site. She's a staff physician, and to get there, she completed her residency in pediatrics with a specialization in hemato-oncology. She obtained her PhD in Paris, and then she came over the Big Lake and followed postdoctoral fellowship here in biochemistry with Philippe Gros at McGill University. A little aparté, Nada was known to raid our freezers at night when she missed some enzymes. And she would come, and the next morning she would say, I promise, I'll bring it back eventually. She did, eventually. She began her career as an independent investigator at McGill in 2003, pioneering a research program in pediatric brain tumors, which is now unparalleled. She is a star. This work and other publications, landmark papers, she's got over 3,000 citations since 2012. She's got over 150 peer-reviewed publications to her credit in the best scientific journals that we all know. We're very happy that she hopped into Montreal in between a couple of trips to essentially deliver her talk tonight, and we look forward to what she has to say. I just remind the speakers you need to stay within the confines here to be taped appropriately.
Merci beaucoup, Nicole. Thank you very much, Nicole, for the invitation and the introduction. Bonsoir, mesdames et messieurs. See, those are just words. Words are made of air, and air makes nothing. I treat patients that have cancers. I am the primary physician for brain tumors. And if we are one day to make any dent in this disease, we have to understand. This is how I actually treat my pain. I don't have kids that have brain cancer, but I treat patients that have this type of cancer, and my pain can go away if I understand better for that to improve and to go further and further. And this is really, it's not me, the star, not at all, not a second. It's those patients and those parents that donate all of those samples, this knowledge, for us to do better with it. And I hope today to convey some of the sense of hope that we're going. So I am a pediatrician, so I tell stories. And the first story is that, you know, one of the things that comes from working with patients that have sick, you don't know what tomorrow is going to held for them. You don't know what's going to happen, especially in a cancer. Oftentimes you win, sometimes you don't. You don't really know. You would love to have a crystal ball. And whenever I look at the movie, I would look at the end. This way I know I'm not going to be surprised because this is something I can control. When I read a book, I read the end. And one thing that happened when we were able to sequence our genome, we thought, okay, this is starting the beginning. We will probably know what is it that we're born with and what is it that we can acquire or not acquire. And I hope that never will go for those who are my age. And so this movie where people get segregated based on their genomic information, I hope we never go this way. This is an amazing tool that came, it's almost a gift. It is a gift in our hands. And this is something that's making us understand faster and better. Is it the only thing? No. It's one of the many means. And what I want to do today is to tell you how we've come to harness a little bit this tool to make a little bit more understanding for the pediatric brain tumors that are also the young adult brain tumors, and probably apply it to other cancers. This is the message that I wanted. It's only one of the many tools. It's a tool that's needed, but it's not going to be the only thing. Knowing our genomic information and the genomic information of the tumor is not going to be the only thing to help us treat. It's one of many ways. It's a good part, but it's not the only way. So the first is how a snail became as a cheetah. I dreamed when I started medical school to be at a time where the reason why I embarked into research is when somebody described to me that there was this enzyme, this thing that worked at very high temperature, the PCR. And this is what allowed us to sequence our first genome. And I was dreaming that one day I would be at that. And I was lucky that the next generation sequencing came at a time where I was ripe to go it. What was it? To sequence one human genome, it took about $3 billion, about 20 years, 200 Sanger machines, one next to the other, and I don't know how many microgram of DNA for one single genome. And if you look at it, if you remember the pixelized picture that you needed to kind of go and focus, this is exactly how it was. Nowadays, with those beautiful tools that we have, we can sequence in 2.5 days, and even 6 hours, if we're lucky to go to the next machine, 96 genomes with a sharpness that's unparalleled, and with a cost of barely 2,000 Canadian dollars, and that's why, because we inflated a little bit. And it's actually predicted to go even less down. This is a revolution in technology, and this is what we're living today. We could sequence everything. We could sequence DNA, RNA. We could look at the methylation, the epigenome that I'm going to be speaking about. And actually, from one sample, from one individual, there are so many different things that we could do, and we are doing them. The problem is we don't know how far those technologies are going to take us. And we, as researchers and whatever, raise hopes immediately by saying we know everything and whatever. People expect an answer. We're doing way better. Look at how we've put a dent for breast cancer. We're putting a dent for melanoma. We're putting a dent in many things. We're doing giant leaps. But when we see our close ones, and when I see a patient, I want already to be there, and I'm not yet there. So with our impatience, we need to be patient because this is probably not going to be enough, and we need more. This is the story of pediatric brain tumor. Anything that's green is leukemia. Leukemia is the first cause of cancer in kids. We can cure 95% of acute lymphoblastic leukemia almost without any problem. Brain tumor are another story. This is a story of the first cause of cancer-related deaths and of side effects in children up to the age of 18, and actually in young adults from 20 to 35 years of age. And this is something, the one that we can cure, and we can cure more and more, I can reassure of that, are left with side effects due to the surgery that's needed to save their life. And I'll tell you later about how we go about that. The radiation therapy that's really mandated in the high-grade gliomas and the other tumor that we treat, and the chemotherapeutic agent that we need to control those diseases. So we treat and we give side effects for those that we can cure. So the aim is, can we do it differently? And those that we cannot touch today, why aren't we touching them, and can we do better? We live by dogmas, and we have. So anything that comes from a progenitor will come and give you all of those cells that populate our brain, and anything that's bad we call the norma. Astrocyte, astrocytoma, neurons, it's medulloblastoma, it's a different story, but this is how we go, and it's pathology. Children are not young adults. This is the first thing. Even a cancer that looks the same, has the same name and whatever, is completely distinct. And this is something that actually, when I started working in 2003, the same treatment were taken from adults to kids for high-grade gliomas. They were not effective. They're not effective also in adults, but they were not effective because when the makeup, the genetic and the molecular makeup between kids and adults are completely distinct, and this is what we've shown. And after having shown that, this is what I felt, it's really, I'm going nowhere. And this is where we embarked on the sequencing, and we were one of the first group, and we hit gold. Why do I say we hit gold? Is that we uncovered mutation in a gene, in a histone gene. This gene is amazing. I put a lot of jargon in my slide. Listen to what I have to say if you want to look from time to time. I don't want to lose you. This histone gene, nobody ever thought that it could be mutated. The TCGA, which is a big consortium that actually sequenced the first human cancer, selected about 900 genes, and this was never part of that because nobody ever thought that this histone gene could be affected or mutated. Why? Because even plants have it. And even they have the same amino acid sequence, which is like extraordinary. If something is as conserved from plants to human, up to the biggest primate, it means that it's very important, and messing up with it is not good. So what does those histone genes do? DNA. In every single cell of our organism, we have the same molecule of DNA, 2 meter 10 long. But you agree that the cell is invisible. You need a microscope. And the nucleus is even more small. So you need to compact this DNA into nothingness. And this is what those histones do. They form what they call the nucleosome. And around this nucleosome, you wrap your DNA, a little bit like a sewing kit, and you have your the fil that's around it, and you completely push it up. And you agree with me that even if we have the same DNA in each one of our cells, an eye is different from a brain, a muscle is different from a skin, and a fetus is different from an adult. This genetic information that's completely compacted needs to be open and to make sense when it needs to make sense. And this is the exquisite role of the epigenome and those histone genes that I told you about, that really open and close our genetic information to make it make sense when it needs to make sense. And on this histone tail that I tell you about, there are marks that you could put, multiple marks. There's something that writes one mark, another one that reads what was written, and another thing to erase it. So we were very, very disappointed when the sequencing of the human genome came and we found out that we had the same number of genes than the mouse, 22,000 genes, and we were different than the mouse. Maybe it's a way we play with our genetic information that's different. Maybe one gene, just based on the fact how we open and close it, how we put it together, can make 20,000 genes, and I'm exaggerating here, but so many more, just based on the fact of how we assemble it. And this is the role of the epigenome. And maybe think about our genetic information as notes of music. The CTGA of our DNA are do, re, mi, fa, sol, la, si. You agree with me, they're meaningless unless somebody put them into a partition. And this is what the epigenome does. With simple notes of music, it creates things like symphonies for Bach, for Mozart, to Adele, to Imagine Dragons, and even rap. And so this is actually what's making sense of our genetic information at the time it needs to make sense. The same molecule of DNA that could give a fetus a brain, a heart, just because it opens and closes the genetic information at the time it needs to open and close it. It's actually the barcode of life. And this barcode is so wrong in those brain tumors. It is singing non-harmonic rap. So what we showed is that we could actually, based on the age and the location of the tumor, know exactly what type of mutation would happen. One, the midbrain, the midline of our brain, you have the cortex, and the midline, the pons, and which is our hardware, whatever, are a problem of a lysine 27. I'm sorry to use this jargon, just lysine 27, and the cortex is a problem of a lysine 36, both of them on the same histone tail. Same gene, the same tail, 2 residues, one on position 27, the other one on position 36. One is controlling the midbrain, and the other one is controlling the cortex. And I know that if it's a 3 to 5-year-old, it's going to be this mutation with this associated mutation. A 5 to 7, the same mutation with another partner, a little bit older, the same mutation with another partner. I could actually guess, even without even knowing the molecular, just based on the age and where it's located, what are the type of mutation. This is a problem during brain development. And this is why those mutations occurred. It's something that went wrong, that got selected during brain development, and it's so tightly fit with it that we need to understand better, and it's a new mechanism. Nobody ever believed that the epigenome and those histone genes could be sick. This is why we got so much citation. This is why the work is, from my lab, now there is, and I'm not bragging, there are more than 300 labs, if not more, working on them because those are tools to understand the epigenome and a new, completely new way of creating tumors. And this way of creating tumors is actually shared with some of the high-grade tumors you see in the adult, and you'll see with that. So for us, what we showed, and we're showing more, is the pediatric and young adults are a story of those two lysine that are wrong, badly read. And it's more than the high-grade astrocytoma. Even other embryonal tumors seems to share their problem with the lysine 27, which tell me that for midline brain development, this is the major problem that's happening, and we need to understand more if we are to treat. The WHO World Health Organization changed the way of looking at tumor. Hooray! So now the K27M mutation that we identify is part of it. And the ATRX mutation that we identified in the young adults are also part of it. Why is that a victory? The victory is that we're not only looking at the slide, at the staining, we're using molecular tools that are so much more precise. Because if we are able to distinguish them, we could potentially treat them separately. Because up to now, they're not responding, and they will respond more when we understand them better. With the help of Millipore, we designed an antibody, and this is something that we put in practice. So no, we don't even need to go to the molecular. We do one staining, and this is across the world. And we could actually make the diagnosis within 48 hours. So 2 marks gone wrong. One, a repressive mark, the lysine 27, that's not speaking to the activating lysine 36. I'll let you guess which is what. And those are the ones that are problematic and are creating the problem. This is what's shaping. This is what putting our DNA, opening and closing it, and those 2 marks are very, very badly wrong. When one disappears, the other one takes over, and the reverse is true. And this yin-yang is something that we're further teasing out. Are there other cancers than those pediatric high-grade glioma and those young adults? So there is cancer of the bone, the chondroblastoma, and the giant cell tumors that have other type of mutation also in the oncohistone. We identified in AML in 2% the same mutation that we see in the high-grade glioma, in 20% of undifferentiated sarcoma, and for a pediatrician, head and neck cancer, 15% was like, for me, a major. And it's important because they offer vulnerability that we could take advantage of because we're understanding more and more. And actually, those tumors in head and neck cancer that have this type of mutation are potentially more amenable to immunotherapy. And we're learning more why that is, and the PFA and ependymoma. We need to create models, and so we've been working on that, and we got the first mouse model, and this is one of my colleagues that probably reviewed the paper that put this as a news and views. It's me. It's really, if you put this mutation too early, it's lethal. Too late, it doesn't work. It's really within a given window of development within the brain that it has, unfortunately, the potential. And we're going more to understand what is the window of opportunity. And we're even using fruit flies, regardless of what Sarah Palin has done. And there is Amel Shawouch here, who is my fruit fly lady and is working on this. And we have beautiful data. It's a beautiful model that's really binary, 0, 1, where you could screen drugs and understand mechanism way easier. And we have no animal control. Nobody cares about a fly if we smash it, if we take its eye. And my daughters have been removing eyes to test the RNA. And they hate me and they will never do research, but that's another thing. We want targeted therapies. We really want that. And not a targeted therapy, it's not going to work if you put one drug. We already identified within the tumor exquisite vulnerability. If we use only that, it's not going to work. The future is to combine, enhance the immune system in some way or another, work on this epigenome that I was telling you about, and work on the kinases, which are like the fire that make your tumor multiply more. And what is the best combination? We need to figure it out by working together, and I'll come to that. The Time magazine knew that not everything is in our genes. Really, epigenome is something that is governing our genetic information, and it's a process that is quite key in cancer, and we're learning more and more about that. And in 2010, when I started, it looked like a beautiful beach like that, but look how it is now. And I'm so happy it's crowded, because when people work together, even if they're working in competition, whatever, it's very good. It's like when you had the first car, it cost like a gazillion, and then now the cost is less and less, and it's really something. So a cure is going to happen because we're more and more, and we're looking at the same thing. And even Einstein wished probably that he would have studied his stones. This revolution is already making its place for the classification of the brain tumor. For the low-grade glioma, it was a mixed bag. I have a low-grade glioma, okay. Then I remove it, I give a lot of chemotherapy and whatever. It looks the same, it acts the same. It's not the same. We know that there are distinct molecular markers. And for the low grade, we're luckier. It's one hit per tumor. And this hit, it goes to a specific pathway, which is a MAP kinase pathway. And why am I bothering you with this? Because there are targeted inhibitors that exist. And we have put out a clinical trials now that target those tumors based on the genetic makeup they have. And it's making its effect. And I'll show you data. Medulloblastoma, it used to be one disease, then 4 diseases, now with 13 distinct diseases. And that's important, because in some of them, I could remove the radiation therapy and only give chemo. In others, I know that whatever I do, nothing's going to happen, and I need to do it differently. So I am adapting the treatment to the tumor and making more sense of the patient's welfare. And this is what we want all. And it's happening more and more. So we really need to integrate those tools and harness them into clinical practice. There are several platforms available, and we really are kind of doing a pilot study that I'm going to speak about very briefly, the profile study, to sequence, to see what are the benefits, to improve diagnosis, to identify targets, and to treat those patients as a collaborative, and I'll speak about that. The challenge is there are so many markers, so many things. The healthcare system, they're like, oh. But just imagine if you maim a child, the lifelong disability that you're creating, and all of the money that you're going to be saving to the healthcare if you just give the right treatment at the right time. For me, there is absolutely no problem about that. Just to show you that it's not something that cannot happen. I'm going to show you this example over here. I don't know if I can show my arrow. Here it is. So this is a child that I saw. I got the 911 page from the neurosurgeon for a kid that was 7-year-old that came not able to use his right hand. His eyes was doing like this. And this is a brainstem. This is the hard drive of all of our body. And within it, there was a 10-centimeter tumor that's right here. Anything that's white from up, actually down to here, is tumor. Completely integrated. He cannot remove it. And it looked like exactly like other tumors we had seen. Remember I told you we could kind of guess based on the images and the age. He took a small little biopsy. The pathologist gave us the answer within 24 hours. It had the problem we suspected, and we had a cure for that problem. Oral pill, twice a day, look, after 24 days. And he recovered his right hand. So even for the brain, a precision therapy can happen. This is a low-grade tumor, but it can happen in high-grade tumor. As long as you identify the fuel, you could find the cure. And by understanding this kid, he came after 2 other kids that received I don't know how many therapies until we salvaged them with this medication. We didn't wait for him, we gave it up front. And this is what we're doing. We're now trying and bring it up front to improve the care. Profile is an initiative that I love. It's precision oncology for pediatric and young adults. It's a pan-Canadian initiative that aims to see how much can we do with the next generation sequencing as a Canadian collective, and how can we integrate with pediatrics the young adults. And we're doing a lot of things. We're taking the tumor, from the tumor, we're trying to get models, the avatars that I spoke to you about, biomarkers to see if we can see if the treatment works in real time and how to adapt it, and the ethics behind it, because genetic information is very sensitive, and a lot of expectations are happening around that. But again, and to build the clinical trials, this way we don't have one child here and there, we have all the children within Canada, all the children within Quebec, part of similar clinical trial where we can see this information and know what works in real time and what will work. And this is what we're aiming. And it's across the board. So I was feeling like a Trekkie going like very fast, beam me up, Scotty, instead of, one step at a time. And then the WHO classification, even though it appeared, this is what it gave. But, those are fights that we'll win. If you didn't believe me that lysine 27 is something important, remember I told you about that? I was looking at a movie with my kids, The Amazing Spider-Man, and I had a free screen, and my kids were like looking at me, what, she's gone crazy like usual. And then even Mr. Spider-Man, even Hollywood knows the importance of lysine 27 trimethylation. Look, it's here. So this is when he's explaining how he transformed. So if you didn't believe me, Hollywood believes me and already integrated it in as part of that. The work that I showed is really a collection of different talents from across the world, people that gave samples, expertise, and whatever. I am the spokesperson, but many people could have spoken about it. The different funding sources, initially I only had some funds, and then they grew. And again, knowledge is important. Research for me is key. I love translation, but zero translates into zero. If you don't have proper research, you don't have proper targets, you don't have proper knowledge, and you could never dream to overcome a disease. So research is part of care. It is to improve care, and it's to help us kind of find those beautiful solutions that we're finding. And thank you for your attention.
Thank you, Nada. I gave you a breather. Our second speaker tonight is Dr. Livia Garcia. She completed her master's degree in medical biotechnology in Italy at the University of Naples. So you might detect a slight Italian accent. She was set to pursue an academic career when she enrolled in the PhD program at the Open University in Cambridge in the United Kingdom. And during her PhD training, she developed a keen interest in developmental biology and its relationship to pediatric cancer, and was awarded her PhD in life science for her research conducted in the Telethon Institute for Genetics and Medicine in Naples. She then moved to Canada to advance her training in cancer genetics with a postdoctoral fellowship granted by the American Brain Tumor Association under the direction of Dr. Michael Taylor. As a postdoctoral fellow in the Taylor Lab, she led the lab efforts towards understanding how developmental mechanisms are altered during tumorigenesis in pediatric brain tumors, with the goal of designing new therapeutic strategies for this disease. She developed the first preclinical model to study medulloblastoma recurrence, which is a major cause of mortality in the patient's population. And then we stole her away. She moved from Toronto to Montreal. She joined the McGill University Research Institute in January 2017. She's an assistant professor in the Cancer Research Program, in the Department of Orthopedic Surgery, which is a little far from the brain, but with complications that are just as important. She's focusing on new developments of a basic research Ewing sarcoma program to tackle the challenge of metastasis and therapy resistance. And we're quite happy to have her here, but she's going to be talking about brain cancer, which was the focus of her postdoctoral research. Okay. Ah, oui. Voilà. There you go.
Thank you. Okay, thank you very much, Nicole, for this awesome presentation. And so I don't have to apologize for my exotic accent right away. Thankfully, I'm going to spare you my French, which is about the level of 5 years old. It's coming. And for now, it's survival level. But hopefully, if we meet again next year or in a few years, I'll be fluent by then. So today, I will focus about a pediatric brain tumor that's called medulloblastoma. So you heard a bit about it from Nada before. And the focus of my presentation will be how the genetic research that we're doing in this type of cancer is translating to the clinic. How can we take the information that we learn from chips, from high technologies, and bring them to the patients, and how this is changing clinical practice. So first, going back to what Nada was mentioning, I always put up this slide because it's quite striking. So we are not usually thinking about cancer in kids, because we think about cancer as something that happens after environmental exposure. You know, you smoke, people drink, people have bad lifestyles, and we all know from television, from internet, that this is bad for you. So this can actually lead to certain type of cancers. But how about kids? You know, they're smoking, hopefully their moms haven't, and same thing for alcohol and other risk factors that are connected to lifestyle. So it's kind of not straightforward to think that cancer is actually the first cause of death in children. So if we exclude accidents and things that can happen without intention, like a trauma or things like that, so cancer is the second cause of mortality in children. So because of my developmental biologist training, I was interested in understanding what is wrong in the genes of kids, of certain kids, that is making them developing this type of cancers when they are not exposed to any external factor that could be causing it. So if we look at the statistics about what type of cancers kids get, they are kind of different from the cancers that adult people get. So you hear that in the adult population, you have breast cancer, colon cancer, lung cancer. Well, the cancers that happen in children are different. They come from different tissues in the organism, and most commonly are leukemias and brain tumors. So my presentation today focuses on medulloblastoma. So medulloblastoma is a tumor of the cerebellum. So this is, I don't know if you can see my pointer. Can you? Oh yeah, here it is. Okay, so look here on the left-hand side of the slide, and that is a normal human brain in the schematic. In the bottom part is I'm getting there. This is not easy. Okay, see that line, the black line that was left over on the slide? So that points to the cerebellum, which is the region of the brain where medulloblastoma tumors form. The cerebellum is that part of the brain that you need for fine motor control. If you enjoy playing a musical instrument, if you enjoy writing, painting, or playing video games, you're using your cerebellum. So when tumors form in the cerebellum, if they stay localized right there, then we call them primary or localized medulloblastomas. And patients are usually looking like the MRI scan in the middle of the slide. I'm going to point you to the tumor, which is right here. See this big ball in the bottom of the brain in the back, which is, I should get a pointer possibly, so I can show you like that. Right, so that is, thank you. So that is a medulloblastoma tumor growing in the cerebellum of the patient. And sometimes kids develop metastasis. In about 30% of the cases, what happens is the tumor spreads from its primary location to another part of the brain and grows. And at that point, the situation is really difficult because I'm going to mess up with this now. Oh, top. Oh, yeah, look at that. Okay, so no more running back and forth. These are medulloblastoma metastases. See this light green part? These are metastases that are growing on the spinal cord of this person, of this patient. This is the spinal column. These are the vertebral bodies. This is the central nervous system that's in the spinal cord. And this is a metastasis. So there is no way the best surgeon in the world can cut it out. You can't. It's spread on the tissue like butter on a hot toast. It's not coming off. We have to kill it by radiation and chemotherapy. So medulloblastoma is not a new disease. It was isolated and identified in 1925 by two very expert neurophysiologists, Dr. Cushing and Dr. Bailey. At the time, we didn't know how to treat them. We would only remove the tumors by surgery and everyone would die. Survival was 0. Then in the 70s, things started to go much better because we introduced radiation therapy, which saved about 60% of the patients. But as Nada pointed out before, the price was huge. The price was huge because these patients were heavily, heavily treated and heavily disabled because of the treatment. Then in 1980 and after that, the introduction of chemotherapy really helped us reducing the radiation dose and improving survival even further. So now we cure about 80 to 85% of these patients. Fantastic. However, there are still issues with certain subclasses of patients. So now if patients come to the clinic and they are suspected to have a medulloblastoma, the first thing they get surgery. So the neurosurgeon goes in, takes the tumor out, and then we look at it under the microscope. And we divide patients in high risk and low risk based on certain features. So the pathologist looks at the tumor under the microscope and assesses if the imaging is showing metastasis, if there is any sign of tumor left that the surgeon left over from the surgery, and if there are tumor cells that are in the fluid that surrounds the central nervous system. If none of those conditions is met, then the patient is average risk. If one of those conditions is met, then the patient becomes high risk, and we treat them way more aggressively. So it would seem that everything, is going really well for these patients, right? We cure about 85% of them, fantastic. However, these numbers don't say the full story, because what happens to the survivors is still needs a lot of improvement. Many children are overtreated because they wouldn't need that much. And many children, despite this aggressive therapy, still can't make it. 85% is not 100, so there is still a lot of kids that are dying from this disease. And the survivors have a very poor quality of life because you're radiating the brain that is developing in a little toddler with massive doses of radiation, and you have to save his life, but that comes with price of lifelong disabilities. So what we thought is that we needed to do more about this and figure out a way to be able to discriminate which patients needed more therapy and which patients could possibly do with less. So this is an example of a case that we had in the Hospital for Sick Children where I did my postdoctoral training. And these are two patients, samples taken from the OR and analyzed by light microscopy, patient 1 and patient 2. Well, you don't need to be a pathologist to be able to tell they're kind of similar, right? It doesn't look like there are huge differences, and you can see just by looking at it. So read about the stories. So the first girl, 6 years old, perfect surgery, no metastasis. She goes on, has radiation, chemotherapy, and thankfully is now 11 years old, no sign of recurrence. The other little girl, still 6 years old, same hospital, same therapy, same surgeon most probably, and unfortunately, that little girl doesn't make it. And the tumors look the same. So what's wrong with it? So this is the question that bothered us for a long time. You know, when people have different type of adult cancer, sometimes you read long reports from pathology that describe the stage 1, 2, 3 with different letters that tell you exactly where your risk is. Well, in brain cancer, this doesn't happen. They all look the same. The pathologists can't tell them apart unless we do something else to look deeper. So we hypothesized that using genomics, we could understand what were the underlying differences between the different cases and potentially understand better what was going to be better for them. I will just go super quickly on a few terms that possibly are kind of confusing. So the gene are the little working units of our genome, and they work all together in networks. So when scientists look at only one gene, only one little unit, that's, we call it a genetic study. When scientists focus on the concerted efforts and the concerted differences between network of genes, so many little units working together, then we call it genomics. So our the analysis that we did is called unsupervised clustering, which is basically a way to make groups. So I don't want you to read that, but I wanted to look at this. Because I'm Italian, I thought that this was a nice fitting example to explain what clustering is. So look at the pizza on the left, and please, don't pay attention to the fact that there is no cheese, so it's not a real pizza, and there is hummus there. Just look at the vegetables. So that is, pretend you have never seen a vegetable in your life, and you can't tell a pepperoni from a zucchini or from an artichoke, one from the other. So those are the medulloblastoma patients. We can't tell them apart. But what we can do, we can ask a computer to look at the differences in how the little genes work and to make groups and to put all the vegetables that have the same features all together and to organize them so that every vegetable is in the same group of vegetables of the same flavor and different from all the other groups. So this is what gets you to the second pizza on the right-hand side, where the tomatoes are all together, the artichokes are all together, and the zucchini are all together. So now we know that on that pizza we put 4 types of vegetables. And this is exactly what we did with science. So these are the real, genomic data where we made groups and we figured out which patients are similar to which based on gene expression, based on how the networks of genes are coordinately expressed in the different tumors. And we identified 4 different subgroups. So we don't have one disease called medulloblastoma, we have 4, which are the Wnt subgroup, the blue ball up here, the sonic hedgehog subgroup, the group 3, and the group 4. Interestingly, this could be just a genetic exercise for, the passionate people that really like to look into genes, but is this relevant? Is this gonna change how we treat these patients? Or, it's just a tag that I can put on my type of disease, but it's not really helping me. Well, it actually does. Don't look at the whole information on the slides. Just look at the prognosis line over here. So Wnt tumors have very good prognosis. Sonic hedgehog tumors have kind of intermediate, and the group 3 tumors have a very poor prognosis. So now we know that while these entities looked all the same, they are different, and they have different outcomes. So that takes us back to the 2 little girls at Sick Kids. So look, first girl who had a very benign outcome was indeed a Wnt patient. So probably she could have gone away with less therapy, and we wouldn't have damaged her brain as much had we known that she was on that molecular subgroup. Patient number 2 instead is a group 3 patient who possibly needs a little more or needs more specific therapy because we are not curing them. Despite the improvements in genetic and genomics, there are still clinical challenges and clinically important questions that we don't know how to answer. So now we have a more detailed understanding of what's going on in these tumors, but we haven't solved the whole puzzle. For example, group 3 and group 4 patients, we know that they mostly die of metastasis, and the sonic hedgehog medulloblastoma patients die of local recurrence. So metastatic medulloblastoma is particularly dangerous because, as I was explaining before, we have no way of operating on these kids. We can't cut the disease away. We need to treat them with drugs, and we need to treat the patients with radiation therapy. So what we noticed in the clinical practice is that in certain cases, patients' tumors, the tumor in the cerebellum responds to therapy, but the tumors, they're spread somewhere else, so the metastases are not responding. And I brought an example for you. So this is the site where a primary tumor was, and now it's empty. So the tumor in the cerebellum did not grow back, but metastases here and here and here did not respond to therapy. And despite chemotherapy, radiation, and a very aggressive protocol, they're there untouched. So what we hypothesize is that there are molecular differences between primary tumors and metastases that are underlying these differences. And we confirmed this in animal models and with patient samples. So they're called Venn diagrams, and they're kind of like summarizing differences and similarities between groups. The gray balls are the genes mutated in the primary tumors, and the orange balls are genes mutated in the metastasis. So the extent of similarity is represented by the extent of overlap between the two diagrams. So only about 10% is in common. So 90% of the genes between primary tumors and metastases are actually different. So it's not surprising that we can't treat the metastases because they have important biological differences. Then we wondered if the same thing could be said of patients that have been treated and their tumor comes back. So when a patient tumor comes back, that is the time when people are enrolled in clinical trials to see if some experimental drug is going to work. To pick the best trial, what the oncologists do is to look at the genetic alterations that were in the primary tumor when they took it out from the surgery, and then treat patients after they received radiation, chemotherapy, and all the other treatments. Well, that is not working too well, like Nada was mentioning for brain tumors. It could work. However, we have to remember that in the time between primary tumor and relapse, stuff happens. The tumor is not sitting idle, waiting for you to kill it. So these are the differences between primary tumors and relapses. So primary tumors and relapses only share about 15% of the genes, indicating that if we decide which therapy to treat the patients based on the genes that we see in the primary tumor, and then we treat patients after they have received therapy, that is not likely going to work. So we need to rebiopsy every patient before they come for treatment at relapse, or we won't be able to do an informed decision of what the best therapy for that patient is. So I just want to summarize what I would say the take-home message of this little presentation is. So on the left-hand side, you'll see those are the 3 discoveries I've talked to you about. So first, we identified differences between primary medulloblastomas, and we defined 4 different molecular subgroups. And this translated directly in clinical impact in terms that now, when a patient is enrolled in a clinical trial, we always look at the molecular subtype, and we make decisions based on that. We don't go blind anymore using microscopes that have been invented 400 years ago. We do better, and we look at molecular differences. And nowadays, patients have been recruited based on their molecular subtype, and Wnt patients are being excluded from very, very high doses of radiation and chemotherapy, and potentially we're going to have less side effects in this patient population. The other 2 discoveries that I talked about are about the differences between primary tumor and metastasis, and the difference between primary tumors and relapses. So how does this translate into therapy? Well, first of all, we understand that we know too little about metastasis. So that is the thing that we are strongly advocating on in the field. We need more samples. We need to be able to look at the metastatic tissue and see what's wrong in it, because we cannot assume that is the same as the primary tumors. So we are trying to put together several centers across the world that have these precious tissues to be able to analyze it and make conclusions with bigger numbers. And the same thing for patients at relapse. We know that patients at relapse have to be treated based on the genetic profile of the relapsed tumor, not of the tumor that was there at diagnosis, because in the meantime, things have changed, and we need to know that. Okay, so I just want to thank all the people that allowed this project. This is not one project. This is about, I would say, another 15 years of, it's about 15 years of work of 45 centers across the world, 1,000 tumors, a lot of hours of flight time for my ex-supervisor, and a lot of work of several postdocs like me, and students and master students. But we all have one goal, that is to cure a rare cancer. And to cure a rare cancer, you have to team up. You have to make big teams and very focused and work together. And I'll be happy to take any questions.
So thank you, Livia. I just want to remind you, you were given little cue cards and pencils. So as you may remember, we have a question period at the end. So if you have questions that you want to bring forward, or we'll collect them as they're made available at the end, and we'll ask the questions of the various speakers in the end. So our third speaker this evening is one of our new researchers, really, really new. She came in last August. Daniela Quayle is a group leader now in the Goodman Cancer Center. She joined the McGill Network. She comes from Memorial Sloan Kettering Cancer Center in New York. Her lab is focused on studying the environment of a tissue and how this impacts cancer progression. She has had a number of projects which she's continuing. She's focusing on breast cancer metastasis, which means when the cancer spreads to different organs, as the lung, the liver, or the brain, as well as studying primary brain tumors such as gliomas. And she's trying to understand new ways of engaging the immune system in order to combat cancer. So this is quite important because there are quite a number of new developments happening relative to immunotherapy and various types of cancers. So Daniela will essentially talk to us about her recent discoveries in this field.
Okay. Thank you, Nicole, for the introduction. I'm really happy to be here today to talk to you all about my work. I study gliomas in adults. I don't do pediatric brain tumors. And I study a particular type of glioma that is very aggressive. These are grade 4, very high-grade glioblastomas. And I don't study mutations, and actually I don't even study tumor cells, but I study everything within a tumor except for the tumor cells. And that's called the microenvironment. Sorry, let's advance. So the human body has more than 30 trillion cells, and each of these cells has the ability to divide, most of them. Don't take that too literally. And an example of this is the skin. We lose 50,000 cells from our skin every minute, and we make 50 million new cells in our skin every day. So a lot of cell divisions that are happening. And every time our cells divide, we need to copy our DNA. And when we copy our DNA, just like you might copy some written document onto a new piece of paper with a pencil, you might make mistakes, and those mistakes are called mutations. So 30 trillion cells dividing every day, and every single time they divide, you might make a mistake. Now, on top of this, to make things riskier and worse, we're also exposed to a lot of carcinogenic agents and mutagens in our lives. So that includes things like pollution, cigarette smoke, obesity, which is another topic that I study in my lab, viral infections and chronic inflammation, aging, different types of preservatives in food, pesticides and other chemical agents you may be exposed to, ultraviolet radiation. So again, there are a lot of risk factors for developing mistakes in our DNA and developing mutations, and we're exposed to these things all the time. But for some reason, half of us will never get cancer, and the other half of us that do usually don't develop cancer until later in life. So that, those kind of numbers really made me curious in the question, why don't we get more cancer? So it turns out tumors are more prevalent than what we once thought, and evidence for this dates back to the 1930s, where people who would be killed in non-disease related things like car accidents, for example, surgeons would find tumors that were indolent and dormant at the time of autopsy, and this is true for pancreas, for thyroid, and some cases in lung and other types of tissues. So the question is, why don't these tumors grow? How do they remain dormant? And this is something that I became interested in when I started my work. So one hypothesis is that while mutations are necessary for cancer, and they certainly are necessary, they may not be sufficient, and there may be another step that is needed in order for a tumor to grow. So some evidence for this. This is an old experiment that was published in the 1980s by a scientist named Mina Bissell, who's published a lot of provocative ideas on the concept of the tumor microenvironment controlling the growth of a tumor. So this is an experiment where the researchers, I just want to see if I can find my mouse. Nope. The researchers injected a virus into some chickens, and this virus causes mutations throughout the entire body of the chicken. And the scientists found that tumors only grew at the site of injection, despite the fact that the virus was a systemic infection. So this was curious. And the scientists hypothesized that perhaps it's the actual physical disruption of the tissue by inserting the needle that causes that tumor to grow in that location. And it wasn't understood why, but that was the hypothesis. And so to test that, the scientists made a small wound at another site in these chickens that were infected with the virus. And this causes inflammation in that new site. And it turns out that a tumor would grow in that new area. So that suggests something important. First, the structure and the actual composition of the tissue and the maintenance of healthy, intact tissue, in other words, the microenvironment, is important to control the growth of the tumor. And if this becomes compromised, for example, if you insert a needle, or if you make a wound, or if you do anything else that can cause inflammation and disrupt the steady state and the health of the tissue, then any transformed or mutated cells that are in that area may be better positioned to grow. So do we have evidence this happens in people? And in fact, we do. So obviously, these chicken experiments are not the same as what happens in human necessarily, but we do have evidence of this. So for example, there are several inflammatory conditions, chronic inflammatory conditions that are associated with cancer incidence and outcomes. And examples of this are colitis and Crohn's disease that are associated with colon cancer. HPV infection that's associated with cervical cancer, hepatitis and cirrhosis, which is associated with liver cancer. Barrett's esophagus and chronic acid reflux is associated with esophageal cancer. Gastritis with stomach cancer. Chronic pancreatitis is associated with pancreatic cancer. And this is just a very small list out of a very long list of chronic inflammatory conditions that are associated with higher incidence of cancer. So again, just like that chicken experiment, these associations suggest that if the microenvironment becomes compromised, so in this case through inflammation, this may enable transformed cells to grow. So I'm using this word microenvironment, but what does that actually mean? What is the tumor microenvironment? And so now I'm switching to brain tumors here for the rest of the presentation. So in here, this is an aerial view of the brain, looking down at the top of the brain in this cartoon, and there's a small glioblastoma that I've drawn on the left-hand side. And these blue cells, if you zoom in, are the tumor cells. And when we think about cancer, a lot of people think about tumors that are a ball of tumor cells. But biology is never this simple. It's never just blue tumor cells that are growing in your brain. And actually, there's a lot of influences from the structure of the tissue in the brain that are important for dictating outcome. So for example, certain cells that are specialized in the brain that interact with these blue tumor cells include things like neurons, astrocytes, and microglia. Microglia are a cell type that I'm going to talk about later, but these are immune cells that are resident to the brain. We also have something called a blood-brain barrier, and this is basically a tight seal that forms on all the blood vessels in your brain, and it essentially stops anything that's floating in your circulation in the rest of your body, like toxins or inflammatory factors or immune cells, from getting in. And the reason for that is because we want to protect our brains. They're a very important organ. We don't want toxins to come in. We don't want inflammation to happen. We don't want our brain to swell. And so the blood-brain barrier helps kind of create a more sterile area around our brain. Now, as these blue tumor cells grow and divide and replicate, they eventually get large enough that they need their own blood supply because they need access to more nutrients and oxygen. So you see increased numbers of vessels, and more recently we've discovered even lymphatic vessels in the brain, which were previously never found before. The problem, though, is these vessels are not normal vessels. They are leaky vessels, and their blood-brain barrier is not intact. And so you lose the integrity of this seal. And now suddenly you get an influx of immune cells, and you're exposed to all sorts of things in your brain that you were not exposed to before. And one thing you're exposed to, for example, is the influx of immune cells that are normally circulating elsewhere in your body and not necessarily in your brain. So you can see, looking at this picture, it's not just the blue tumor cells, but it's a lot of other factors that are surrounding that tumor, that are interacting and communicating with that tumor, that may affect the outgrowth of that tumor and the outcome for the patient. So getting back to glioblastoma, this is a very difficult disease for patients. Average survival for patients with glioblastoma is only 4 months if you don't do any interventions, and with aggressive treatment interventions including surgery, radiation, and temozolomide chemotherapy, these are standard of care, this really only extends to about 15 months. Now, the standard of care treatments of radiation and chemotherapy target cells that are dividing quickly, and so these are your tumor cells that are dividing rapidly. And the bottom line is they're not working. 15 months, although is a victory for this disease, is really not improving anybody's life. So my lab is interested in asking, can we identify new therapeutic targets that really harness the ability of the microenvironment to affect the way that the cancer grows? And so the analogy that I like to use is a plant that you might put into your garden in the spring. So let's say you want to plant a tomato plant in the spring. What do you need? You need the correct soil conditions. You need to think about whether you need any pesticides, how far away you need to plant your tomato plants from one another. You may need different types of additives to your soil, like fertilizers, in order to help the plant grow. You need to think about the light conditions of that tomato plant. You may need to think about how often you need to water that plant and how much water you need to use. All of these different things you need to think about so those soil conditions are just right. And if you take away any one of these soil conditions or these needs for the plant is going to die. So you can think about your tumor as this plant, and you think about the brain tissue as the soil. And so my lab is focused on how can we change the soil so that that plant dies. So macrophages are a cell that we focus on in my lab. They are the most abundant immune cell in glioma, and in some cases can comprise up to 40% of the bulk tumor. So this is a substantial proportion of the tumor. We know in patients and also in our mouse models, macrophage numbers tend to be higher with elevated glioma grade, with the highest infiltration of macrophages in the glioblastoma grade 4 tumors, and they tend to be associated with more aggressive disease. But as I mentioned, macrophages are an immune cell type, and immune cells are supposed to be the good guys. So why are these associated with poor outcomes? So in order to understand this question, we need to understand what macrophages do. So macrophages are what are called professional phagocytes. And phagocytosis is the process of engulfing or eating any foreign pathogen or virus or also damaged tissues within our body. So in a way, they're the housekeepers of our tissues. And macrophages come in two different flavors. There's the fighter and the lover. The fighter that I have on the left side of the screen are the macrophages that encounter a pathogen and they want to defend our tissues and they want to keep us healthy. And then once we've overcome that infection or that tissue injury has been resolved, we need to put the brakes on that fighting reaction, and we need to tell those macrophages to calm down, and that's where the right macrophage comes in, the alternatively activated, the lover macrophage. Now, unfortunately, glioblastomas train our macrophages to be the lover type of macrophage. And so that basically kind of puts the brakes on all the immune reactions in the brain and gives the brain the soil that it needs in order to grow. So it doesn't have the same type of insults coming in from other immune cells, including macrophages. So this is a problem. So given their high abundance and their pro-tumorigenic behavior in brain tumors, when I started my postdoc, I was interested in asking if we could target macrophages in glioblastoma to improve outcomes. So what happens if we target macrophages? Just for comparison, I want to show you some data here. This is the effects of radiation therapy in the model that we use. This is a mouse model. And keep in mind here, this is a Kaplan-Meier, where every time you see a step down, this means that a mouse is dying. And when I mean that a mouse is dying, I don't mean it's actually dying, because in research, the endpoint in our mouse models is never death. We treat our animals with respect. We make sure that they're comfortable. So every time it's a step down, it actually just means that the mouse is becoming symptomatic, and we euthanize it humanely. So in this tumor model, you can see that treatment with radiation, shown in gray, does provide a modest survival benefit. So the mice do live longer. And again, radiation is targeting quickly dividing cells. So this is a therapy that's really meant to kill tumor cells. Now, if we look at the effects of targeting macrophages, so this is a drug that does not touch the tumor cells. It only targets the macrophages in the brain in the same model. Now we have 44% of our animals surviving till the trial endpoint of 26 weeks. And there's a substantial difference now between the effects of the macrophage-targeted therapy shown in red and the control therapy, which is essentially a sugar pill. So just to show you by MRI just how dramatic that is, so on the left side of the screen, this is the effects of the control drug. So again, the sugar pill. before treatment with the controlled drug, and then on the right side of the screen, it's a similar sized tumor before treatment with the macrophage-targeted therapy. These tumors look quite similar, and they are very similar because this is a very controlled mouse model. But if you treat this tumor on the left with the sugar pill, after just 1 week, there's a 500% increase in the volume of this tumor. Whereas if you treat the tumor on the right with the macrophage-targeted drug, you see a 15% decrease. So if you really compare those tumor sizes after 7 days, there's a quite dramatic difference. So how does this work? First, we wanted to know, are we changing the number of macrophages that are present? So for example, is this drug just depleting them all, and because they're bad macrophages, that's resulting in better outcomes? But it turns out that's not the case. So if you look at the left square here in the vehicle control treated tumors, you see lots of red little macrophage dots. But after 7 days of treatment with this macrophage-targeted drug, you still see plenty of red macrophages in these tumors. So it's not that the numbers are changing. So then we wanted to ask, are we changing the behavior of the macrophages? And so we took macrophages from our control-treated tumors and the tumors that were treated with that macrophage therapy, and we looked at what genes they were expressing. And we found that a lot of different genes, shown in blue on the left of this graph, were being downregulated, whereas fewer were being upregulated. And a lot of the downregulated genes had to do with that alternative activation. So in other words, the lover macrophage was being silenced and allowing that fighter macrophage to kick in and start defending the tumors, or defending our bodies by killing the tumors. So if this is true then, and it is the fighter macrophages kicking in, we would expect to see enhanced immune defenses if we looked in the tissues. So again, macrophages are phagocytes. They eat things and they keep the different tissues clean in our bodies by ingesting pathogens and damaged tissues. So can we see evidence that they're also ingesting the tumor? And what we found was that this is exactly the case. So on the left here, this is an immunofluorescent image. The blue dots represent all the different cells in the tumor, and the red dots represent a dying tumor cell. And you can see the macrophages, which are outlined in green, literally engulfing the tumor cells and cleaning up that tumor. And we know that this is statistically significant if we quantify this. When you treat with the macrophage-targeted drug, shown in the middle here in red, you see that there is elevated phagocytic capacity compared to the control animals, and it's also even higher than when you treat with radiation. So this is great. Have we cured glioblastoma? Obviously not. It's not that simple. So if you look back to these survival curves, you'll notice that although 44% of our mice survive, and that's a great statistic, 56% of them also die. So what's going on? So this is an MRI showing that there's really just an empty space in the brain of these mice that have survived, but if you look at the mice that have died, and essentially that's every step down in this Kaplan-Meier curve, you see that there are these giant tumors that are eventually rebounding. So we want to know, how is this happening, and can we stop it? So to simplify this, I'm going to use a schematic rather than going through all the data, but I just want to recap some key points to take you through it. So first, there's two types of macrophages. We have the fighters on the left, the lovers on the right. The glioblastoma favors the lovers, and so that really is what enables them to grow in some cases. But we've identified this drug that can essentially stop this romance that they're having, and this allows the fighters to then thrive and fight off that glioblastoma. Now, over time, some mice don't survive despite that you have this positive interaction between the macrophages and the tumor cells, and the question is why? And if you remember at the beginning of my talk, I told you tumors are more complicated than just blue tumor cells, and that's true, but they're also more complicated than just tumor cells and macrophages. There's a lot of different cell types that are involved. And in fact, after treating these tumors with the macrophage-targeted drug for a long period of time, we see an influx of T cells. So this is a third cell type now coming into the microenvironment. And these T cells communicate with the macrophages, and they tell the macrophages to secrete this protein called IGF-1. And IGF-1 is exposed to those tumors. And it turns out IGF-1 helps the tumors to grow. So over time, you get this feedback loop that essentially helps the tumor to regrow, despite that this drug initially had effective effects. So next we wanted to ask, what if we target macrophages and IGF-1 signaling together at the same time? So to do this, we went back to our mouse models and we enrolled our animals with the high-grade glioblastoma on the macrophage-targeted drug. And after a period of time, when the T cells would come in and they would stimulate those cells to produce IGF-1, we would treat them with another drug that basically blocks IGF-1 signaling, specifically through PI3 kinase. And what we found is that now, if you compare the red line with 44% survival when we're only targeting the macrophages, now we've improved this to 91% survival. So we have a significant advantage if we block both mechanisms at the same time. So what I've hoped to have convinced you of today is that cancer is more than just tumor cells. Mutations are certainly important, but also it's more than just mutations. And you really need to have a microenvironment that's permissive to growth in order for a tumor to actually grow out. And we've identified that we can possibly target both tumor cells and the microenvironment to really optimize benefits. And we're doing this now in patients who have glioblastoma. They're being treated with macrophage-targeted drugs, including CSF1R inhibitors, which is the drug that I talked to you about today, in combination with radiation and immunotherapy. So this is something that's now being translated to the clinic. And although I've talked to you about macrophages, we can't forget that there's a lot of other cell types up on that picture behind me that potentially have a lot of promise in order to develop new therapies that may be effective against the tumor. So with that, I would just like to take a moment to thank people who are involved in this work, the majority of which was from my postdoc when I was at Memorial Sloan Kettering in New York City, and that's now translated to the work that I'm doing in my own lab. So I'd be happy to also take any questions during the question and answer session.
So thank you, Daniela. Very instructive information. Normally, at this point in time, as you probably remember, we usually have a patient. And Nada, who's a clinician, had identified a few patients. But unfortunately, these patients, of course, are very sick. And none of the ones that had been sort of approached was able to come and tell you a little bit about their disease. So we thought for this one time, what we would do would be to ask Nada to tell us a little bit about, in her case, these little people that get brain cancer and how they deal with the disease in a profoundly touching way.
So I can't actually hope and convey just me what people and patients face. But I could tell you a few examples. And again, the fact that not a lot of people wanted to come forward, it means, you know, when you survive a cancer, you have ownership. You feel like, I don't know if any of you that are here kind of face it or had to face it. We know of people. It's a lot of them, once they overcome this fight, become empowered and invigorated by that. It's completely different for brain tumors, because brain tumors occur in the brain, and anything that affects the brain maims you. So instead of kind of coming with ownership of your survival, being empowered, being, you know, almost proud that you were, those are like very, very hard experiences that nobody can, unless you live them, you can't reproduce them. They don't feel the same way. They feel diminished. And actually, there's been a lot of studies that show that for high-grade, so for the patient with brain tumors that survive, for the high-grade ones, they're really excluded. They're excluded from school because they're viewed as not as normal based on the handicaps that they accumulate. It's in the growing area of the brain. Already the tumor made damage. Some of them have speech issues. Others have ways of walking. They, even if they are potentially mentally and cognitively fine, the exterior appearance of radiation therapy that creates alopecia, that creates shorter stature, that make them different. And this difference is already flagged. And we live in a society when you flag something, it's very hard. But this survivorship is changing. And mark my words about it. The same way we were used to do it for leukemia, where we used to irradiate a lot of those children and give them long side effect, we now know better. And mark my words, first of all, we've adapted the therapies. The radiation therapy is given way more, in a way safer. We're getting even some proton in some instances, and children are being sent elsewhere because we don't have yet the capacity to give proton in Canada, but we're sending them, and the government is agreeing to do that, just to kind of spare them and spare the normal brain when it's possible. We're adapting the chemotherapy. You saw what my colleague Livia was saying. It's to the treatment, and we're making it better. So I'll tell you about 2 examples and my ideal example. The first example is a low-grade tumor. So a patient would come to the emergency. Why? Because either they had a lot of headache, a lot of vomiting, and then it's been ongoing for some time. Sometimes they will have some eye movement and whatever. The diagnosis will be made very fast, and an imaging would be done. An MRI will show the presence of the brain tumor. The first person, the very, very first person to come would be the neurosurgeon, because this is the first life-saving. And we'll assess, is there a way, so we always make decision in a calm way. If there is an emergency, they shunt, meaning if there is like a lot of pressure on the brain, they decrease the pressure in order to come after that to the very best decision to be taken. And then we, as a group, see, okay, can we remove it? Does it look like a high grade or low grade? Remember what I told you, should we do a small little biopsy or should we remove in bulk? Because this is the way to go. And oftentimes when the tumor is in an area where we could safely remove it totally, this is the first step that's favored. Because this is the first, if you could, sometimes for the low-grade tumor, when you remove them completely, there is no need for other treatments, 0. And this can happen. And we'll analyze it so the tumor gets removed or debulked or whatever, and enough material will get an analysis. And the analysis is twofold. So what we do is the pathologist will look and will do what needed to be done. And if the patient agreed to do research for the research and the parents, we do more on that, what I told you. And we complement as needed. Already we have amazing people looking at it. So as needed, we complement diagnosis. And we say, okay, this looks, there is a target here, there is there. And we adapt the therapy as a group. And the patient will either just nothing, radiation and chemo, or a pill. And after that, we follow those patients, and we try and have them go to school. So if it is an oral pill, they go to school. They don't. Some of the treatment we give for the low grade, we really adapt them that this way the kids have only to come to get once a week their therapy, and they go. We adapt it based on the time that was a school, and they could go back to school as much as possible. But the high-grade tumor is not as possible as that. They have to undergo radiation therapy, and they have to undergo high-grade chemotherapy, so it's about 9 months to a year of misery, which is not trivial in the lifetime of an individual. Again, we are gaining more and more ground. We're curing more and more children. What we now want to do is to aim that survivorship comes at a lower price. What we're seeing is that for the children that, not all of them, you never generalize. Some of them I see, they've become actually lawyers, doctors, they can keep contact with me. We really reinforce, we have support groups that we connect them with other children and other young adults that kind of went the same path, and to have for them. We, for those that cannot have their hair regrow, we're trying to do implants, and we kind of go with the wigs, whatever. So we're really trying that the hospital becomes not a house or anything, but that their experience is as positive as could be. And we're fortunate in the pediatric realm to have the possibility of having other people that come and help us this way. And we're trying that this type of support be the same also for young adult and adolescent. And I can't speak for the adult world, but it's also something that's being considered over there. So again, it's very grim, but not always. There are positive and there are negatives. And I think we're moving forward. I would like to take giant leaps. We're going like in big steps, but we're going in the right direction. And I'm happy to take any questions.
So I would ask the speakers to maybe come to the podium in the front, and the volunteers will be coming down the aisles if you have cue cards with questions on them. Please make sure to give these questions, and we'll be asking the questions of the... I would also like to let you know that we are on a number of platforms that you can follow us on. You see our visual crew here. We're on TV. So within a week or two, the whole presentation will be available on our website. And if you want to revisit this or send your friends the link so that they can essentially see this presentation, please do so. We also would like to know, what would you like to hear about next year? We are planning another series for next year, and there's a survey card in your little package, and it's usually quite interesting for us to read what you have to say and to read what you want to hear about. So the first question I have is it possible today to ask for targeted therapy towards a growing benign brain tumor instead of surgery for an older person? And if yes, where to go and who to ask?
For targeted therapy, you need to know what the target. So it's not one target. It's unfortunate, but there are many different targets based on different tumors. And again, even in an old patient nowadays, neurosurgery, it's never easy, but it can be done in an awake person. And if a neurosurgery can remove the tumor, even if it's a benign tumor, this is a first step. What I showed you for the BRAF, it's for the tumor that it's a very unique mutation and a beautiful response. And again, the kid needed to have a biopsy in order for us. We don't have yet other means. We're working on trying and finding either in body fluids or anything, a way to kind of find surrogate instead of going and biopsying the brain to see that, but we're not there yet. So it starts with a small biopsy. It could be done in 2 steps, but if a tumor can be removed safely, I personally would advocate instead of getting a targeted therapy. Because for now, we could put them, and we know they respond. But we know that for a lot of those that respond, the moment we stop this targeted therapy, the tumor comes back. So it's a double-edged sword. And this is something that needs to also be taken into account.
So Daniela, you talked to us about inflammatory conditions. And we have one person in the room, and probably many others, wondering what could be the inflammatory conditions in the brain?
So it's a great question, and the brain normally is supposed to not have inflammatory conditions. So if you think about what happens when you cut your finger and you get an infection, it swells. The brain is not meant to do that, and that's the purpose of the blood-brain barrier. So generally, you're not supposed to have that type of inflammation in your brain. And that's why things kind of go wrong in the context of cancer, because the blood-brain barrier is disrupted, and now you've got all these immune cells and inflammatory factors coming in, and the brain doesn't know how to deal with that very well. So that would be my answer to that.
So in the brain, even if you don't have the inflammation like Crohn's disease or rheumatoid arthritis or other things, however, as we age, and glioblastoma is actually a disease or cancer of the older patient, we get strokes and mini strokes. So when you get a stroke, you stop the blood flow to a specific area of the brain, and you're asking those specific area actually to regenerate. And this is part, and it so happens that it's in the area of strokes that you get the glioblastoma. Is there a correction? It goes with the injury component. It's a different type of inflammation. It's more injury due to blood flow not coming there. And there may be a link between this and the glioblastoma.
So Livia, someone is wondering.
There's another question. No, you don't have a mic, though. I can speak. I've had 2 craniotomies. I've never had brain tumor, but I've had melanoma cancer, and I've had tumors that metastasized to my head. On 2 separate occasions, I had blood in my head, and the blood was caused because I had a blood clot in my right leg, and I was given a fragment which sped up my blood and basically caused seepage in where the suture was put in my head. And the second time, I actually had water in my head, and basically that came from, once again, after surgery, and somehow there was water in my head. In both instances, I had to take steroids to basically remove the water in the head.
So one of the things, again, cancer, it's not all injuries, it's not all strokes, it's rare. Thankfully, it's very rare. It's unfortunate that those, that get it, but it's not because you have chronic inflammation that you'll get cancer. Your risk is higher, but not so much higher, actually. And it's really, so there are so many different traumas that happen, so many different injuries, so many different people that have strokes. And probably barely, it's already too much, but barely 2 or 3% will get, and I'm being very generous with the 2%, actually.
And by the way, the medication, the steroids, was only for 2 or 3 weeks. In other words, they were simple challenges that were resolved through simple medication, not simple medication, but a simple medicated process.
So, Livia, we're wondering if the work that you have presented, would it be applicable to adults as well, adult tumors?
For the adults, brain cancers like the one that Daniela described, we already talk about very high grade tumors where we know which one are the bad players. So we have morphological ways to tell the bad adult brain tumors from the intermediate and the better ones, so to speak. In the pediatrics, we didn't have it. So that type of approach was really needed, where just looking at things was not giving us the right answers. In other adult type of cancers, we pathologists have the means to tell tumors apart and assess the risk to a certain extent by morphology, and then as with more detailed analysis, we can understand what differences are between different subclasses of very high-grade gliomas. Certain types have certain mutations and they behave a certain way, and others have different mutations and behave other ways. But overall, in adult cancer, that is well known, and we are very thrilled to have been able to do this breakthrough findings also in pediatrics.
Daniela, do craniotomies and radiation affect the integrity of the blood-brain barrier and consequently the tumor microenvironment?
I have never performed either of those things on a patient, so I would defer to Nada.
I've never performed, so no. Otherwise, we would never be able to do. It's an irritation, but it goes away. It's not gonna. There are so many people that get surgeries and brain surgeries and whatever surgeries and craniotomies. It's something that we recover from, and your blood-brain, you're not more prone to getting more toxin or anything, so it's not affecting your blood-brain barrier.
So along the same lines, can concussions cause brain tumors? The Canadians have been eliminated now, so we can talk about concussions.
So concussion causes Parkinson's because it's the frontal lobe and it goes, and neurodegenerative disorders. They've not been associated with increased risk of cancer. So, and the concussion, the brain, 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 the but it's the relationship between the Parkinson's disease and the dementia have been very clear.
So I'm not sure which one of you can answer this question, maybe all three of you. Are there different types of brain neurons, or are all brain neurons the same?
I wish they were the same. It was so easy, no? So go ahead.
The short answer is no, and that all the cells that are interacting in the brain and communicating in the brain are very complicated, and the interactions are unique and context-specific as well. As you could imagine, based on how a brain works, even if you just have a very basic understanding, the brain is very complicated, and so that's just the short answer of it.
What is the rate of recurrence in cancer for children treated with brain cancer?
So it depends on the type of brain cancer. So for the low grade, if you remove it completely, it's almost close to, I would say, 5% to be very conservative, and even lower. For the high grade, the medulloblastoma, probably 85% are okay, and it comes back in 15%. This is what we see. And for the other, the high grade that we can't treat, unfortunately, even when you remove it, it comes back. The high grade glioma, the glioblastoma, even if you remove it completely, it comes back. And despite what I've told you and what we know about it, we don't yet know how to touch it.
I think we have a question here from someone who has the potential for a PhD. What are the triggers for genetic changes from primary tumors to recurring tumors? We're just correcting final exams now. This is the kind of question that we ask our students. Let's see what you can come up with.
Well, there are two different aspects to it. So in certain cancers, not all the tumor cells are the same. So in certain types of medulloblastoma, 90% of the cells have a mutation A, and there is 10% has mutation B. And A grows super fast, and then chemotherapy and radiation wipes them off, because chemotherapy and radiation target the fast-growing cells. Then cell B is a bit more, lazy and stays there, grows less, but is the one that's going to win the marathon, because it's not as sensitive to radiation and chemotherapy as cell A. So it's not so much about what radiation and chemotherapy trigger in terms of triggering new mutations or causing specific types of mutations that are worse than the previous one. It's more like chemotherapy and radiation are only killing certain types of tumor cells, leaving the others, the underdogs, kind of like, like an open field to grow back and form recurrences.
Just to elaborate a little bit more on that, Daniela made it's a seed in the soil. When a tumor spreads, metastasis is very ineffective, thanks God, actually, that it's very ineffective. You need to go from, let's say, a breast, you need to go and live in the bone, you need to go and live in the brain, you need to go and live in the lung, that are completely different. It's as if you're an immigrant that was used to living like a Bedouin under a tent, and you come to a place that's completely industrial, or other places, there is an adaptation period that's huge. And oftentimes, the cell will die because it cannot adapt. And unfortunately, the ones that stay and can adapt are the ones that are that go. And this is why now what we're facing in cancer across the board is really there are the brain tumors that really we can't treat because we can't remove, and those are a problem. But those that we can remove, and then the one that went and spread, and they learn how to spread and how to live elsewhere. And those are the ones that are causing a problem, and we need to understand how they learned it and how to remove this root.
So the next question can be either Daniela or Nada. Why is anti-angiogenesis or anti-angiogenic therapy not a good strategy for glioblastomas?
So anti-angiogenic therapies have been ineffective in a lot of different cancers. They're very complicated, and there are a number of different theories on why that might be the case.
So anti-angiogenic therapy is targeting new blood vessels that grow. So if you can stop the growth of the blood vessels, you'll stop feeding the tumors, you'll starve the tumor, and it should die.
Yeah. So this is just an example of a scenario where you could imagine anti-angiogenic therapy not being effective. If you have a tumor that's growing and it's recruiting new vessels and you want to hit it with an angiogenic therapy in order to block those vessels, the result is that the tumor is then going to lose oxygen and become hypoxic. And hypoxic is actually bad for outcomes. It makes cells more aggressive, and the cells that are able to survive in those low oxygen, poor nutrient-rich conditions are tougher cells. And so you're basically selecting for the tough cells, and those are the cells that end up dividing and growing, and then it's harder to target those cells with other treatments. So that's an example where you can conceive that those anti-angiogenic therapies may not work.
Are there any biomarkers for early diagnosis of brain tumors by liquid biopsy that are on the market?
So a biomarker is something that you could use as a surrogate to see, do I have a tumor? This is a dream, is like something that you could take. You could take a blood sample, and from the blood sample, I have a risk of developing a colon cancer, a brain cancer, whatever. Brain is the hardest. Up to now, in the plasma, it's very hard to identify anything that's reliable and going, where there are a lot of efforts ongoing. For other cancer, there seems to be some ideas, some new, but it's still very nascent. So I wouldn't say that there is something that's robust to predict if you're going to get a cancer. What we're seeing is for solid tumors, meaning that cancers that are outside of the brain, and you could do, if you study the tumor, the genetic makeup, you could potentially design a signature that you could follow in the blood. And a lot of studies now are using this as a companion to try and see, is it really something that we could put into clinical practice? Can we detect if this tumor is responding just based on the blood sample, and can we detect that it's coming back on the same blood sample done sequentially in a patient? So it's not a dream, it's something that's going to happen, but it seems to be very much harder in brain tumor, probably because of this blood-brain barrier. Not a lot of things are circulating outside.
What about the CSF for patients?
That's an excellent question, but again, this is something that CSF is beautiful. It's showing a lot of those markers. At the same time, a lot of adults are different than kids. In kids, for some tumors, it's part of the standard of care to take CSF, but for other tumors, it's not part of standard of care. So we're putting in a pilot study to kind of assess that prospectively, and maybe it will become a standard of care.
So the next card says, very insightful talk from all speakers working in the same hospital as the MUHC, but we'll add the Goodman Cancer Center to that. For different mutations, you are heroes. You're real heroes.
The families and the patients.
Yes. For the different mutations that were mentioned, when do they occur in the respective cells? Before, after, or during embryogenesis? And is it different in different types of tumors?
So yes. So for again, kids and adults. In kids and in young adults, those are developmental diseases. It's during the development of the organ, when we can't yet pinpoint it. The studies that we did for the glioblastoma, the one that I spoke about, pinpoints to time during fetogenesis. And again, we need more data to go with that. Is it something that's it's exposure? No. It's really, unfortunately, bad luck. Darwin is here, we're toying with our genome. We're going, this is how we evolve. This is how we evolve to actually combat infectious diseases. Is that our genetic information is not stagnant. We're able to evolve with our environment, to adapt to it, to grow with it. And unfortunately, sometimes part of this evolution especially in kids and young adults, is generating those cancers that we treat. I know it's scary when you hear Daniela speak about the trillion cells that are dividing, that each time we're shifting from the original blueprint. You know, if you look at the statistic, we're surviving so much longer. We are living longer. We are living better. It's not, even if there's the incidence of cancer, we weren't meant, you know, if you look at things, we were meant to kind of be born, procreate, and die. Now we procreate later, and we die much later, and hopefully we'll die much, much later in a good shape. And it's the cancer that we get as older patients are way more indolent, small, you know, less hard for a lot of them to treat. So it's scary, but at the same time, there are answers, and we're providing more and more answers to that. In kids and young adults, it remains something of a bad luck that unfortunately, for now, we cannot predict and we cannot prevent. And I hope to be wrong and that things will be able to come up and we'll be able to prevent it, but that's not happening.
Also, if there's any way I can make that 20 trillion cell thing seem less scary, if you have differences in your DNA, it can also lead to good things too. This is how you have survival of the fittest in evolution. So this is a very important part of our genetic makeup, is to have differences between individuals. And sometimes, unfortunately, that leads to bad things, but other times it leads to good things. And again, that's how evolution works.
So the last question, are there any clinical trials being conducted in Montreal, particularly on astrocytomas?
There are gazillion, not gazillion, I wish there were gazillion trillion. So just to give you, so for adults, yes. Young adults, of course. The moment you're above 18, you have access to immunotherapy, combination drugs, and whatever. Are these clinical trials associated with the genetic makeup or the biological marker? More and more. Not as much as I would like it, but more and more. Kids are a different story. Industry is afraid of kids. Why? Because they have a potential of harm or being harmed for the longest time. It's a small market, and people are not interested. If you have a big drug, you don't want to lose it if you have a kid or two that have major side effects. So industry is scared. I just had a phone call yesterday, actually, with buyers who bought a small company by the name of Loxo. And Loxo has a beautiful drug that targets one kinase, and it's actually 5% of infants that have the high grade. And it's almost, you remember the magic wand that I showed you? This is a magic wand for those type of tumors. And they're scared to death because buyers never, ever went into pediatric oncology. So having clinical trials in kids is not as easy. We're opening them because now there are incentives from the FDA, from the government, for pharmaceutical companies to open a pediatric arm. They're even coerced into some places. And the profile study that I told you about has a specific arm where we're kind of batting with advocacy group and others, and there are some people here that are part of those advocacy groups, to go and have our regulatory authority, Health Canada. It's a major blocker because it needs to protect, but sometimes it's overprotecting and not allowing access of some drugs to kids just because there's not enough data, of safety data. The second, me as a pediatric oncologist, other people, you don't want to deviate from the standard of care just because there's something new. How convinced you should be, how to convince them. And putting those treatments at the forefront and not at the time of relapse where the tumor is way more complicated. All of those are hurdles that make the number, I'm giving a long answer to a short question, the number of trials not as much, but they're increasing and there's a common will. And actually by working together within Canada, within North America, through those big group trials, we're gonna make it happen.
So I would like you to join me in thanking our speakers tonight. I thank you for coming. We have a fourth forum on Wednesday, May 16th on stem cells and cancer stem cells and what's happening in this field in terms of new developments and new treatments. Thank you for being here. Fill out those surveys and send it to us or give it to us. And thank you for coming. Have a safe evening driving back or getting home.